Hello, this is Daniel Shimel presenting my BIO201 modeling project on the phototransduction of the rod cells in the retina. Rods and cones are photoreceptors that detect photons. Photons are the unit of visible light. These are common units of measurement. However, light is measured like this, in wavelengths. Not all light is visible, but this is a representation of the spectrum that is visible. So photoreceptors, there are rod cells and cone cells. Rod cells are responsible for detecting light, whereas cone cells are responsible for the detection of color. For the purposes of this project, I will be focusing on rod cells, which specifically detect the presence or absence of light. So in this representation, we have a rod cell, which is kind of like a magnet that detects light even when there's very little. So rods contain special compounds called visual pigments that are flattened into discs, and then discs stack to build the outer segment of a rod. Within discs are rhodopsin, which is the visual pigment. Rhodopsin consists of the protein opsin, which is bound to the pigment retinol. So this all happens here in the retina. And here we have a closer look of our rod cell. And then here we have a closer look of the discs. Within the discs is the rhodopsin molecule containing the opsin and the retinol. So photoreception has a resting state in darkness and an active state in light. The outer segment of rod cells contain chemically gated sodium ion channels. So in darkness, the channels are open and kept open by a high energy compound called cGMP. So here we have a close-up of our resting. So we have our discs here and our plasma membrane with our channel that is kept open by cGMP. In darkness, when the channels are open, the membrane potential is -40 millivolts. The inner segment of the rod cell also pumps sodium out of the rod via the exchange pump. As pictured here, we have our sodium going into the rod and exiting it. So retinol, the molecule that we mentioned earlier, has two possible configurations, the 11-cis retinol and the 11-trans retinol. When the lights come on, our active state kicks in. So the curved 11-cis retinol straightens, becomes a line, and then becomes 11-trans retinol and activates the opsin molecule. So here we have our photon, and it straightens our retinol right here, which activates the opsin molecule. So the activated opsin molecule then activates the enzyme transducin, which then activates another enzyme, photodiesterase, or PDE, which breaks down cGMP. So we have here a visual representation of our activated opsin molecule, which activates the transducin, which then activates the PDE, which breaks down the cGMP, which keeps our gate closed here, and therefore the sodium is deflected. The absence of cGMP causes the chemically gated sodium channels to close, and the reduction causes their membrane potential to hyperpolarize to -70 millivolts. When a membrane potential hyperpolarizes, it becomes more negative. The vice versa is true, so depolarization is becoming more positive. So this hyperpolarization causes the rod cells to stop releasing neurotransmitters to the adjacent cells. And the reduction of neurotransmitters signals to the adjacent cells that the photoreceptor has absorbed a photon